Sorry for the inconvenience.
That's what I do. Listen, that one I do.
Good morning, one and all. Good morning, one and all. I welcome you all to the webinar series on the occasion of World Pharmacist Day. We have with us Dr. Anil Babu Katiki, and we have as our esteemed speaker here. A warm welcome to you, sir. I welcome you all to this webinar. And today's topic is stem cell research and its fundamentals, conducted by our college, Aditya Bangalore Institute of Pharmacy Education and Research. I request all of you to please mute yourselves. Thank you. To speak on this topic, we have our esteemed speaker, Dr. Anil Babu Katiki, who is a biomedical scientist of the company Hela Cells International Private Limited in Bangalore, Karnataka. I welcome you, sir, once again. Thank you for speaking today. We have with us our beloved principal, Professor Suchitra D, who has been a constant help and support for this webinar. Thank you, ma'am, for being here. I welcome all of you who had been a part of this webinar. Thank you all for being here once again. And last but not the least, I welcome all the students out there to this webinar. I hope you learn from this webinar. Thank you once again, all of you. Now, Dr. Anil, I would like to speak a few things about you. I'm very privileged to talk about you. Dr. Anil is an enthusiastic stem cell biology researcher who had completed his master's degree in stem cells and regenerative medicine from University of Chester, UK. He had completed his graduate degree in Doctor of Pharmacy in Aditya College of Pharmacy, Bangalore in 2019. As part of his role, he worked on multiple cell lines utilizing cutting-edge molecular techniques. He had worked on induced pluripotent stem cells and its application on neurodegenerative disorders. He conducted many successful experiments during his time of graduation, which led to significant future studies. Currently, he is a freelance scientific content writer and part-time biomedical scientist working in the company Hela Cells International Private Limited. His master's thesis was induced pluripotent stem cell transplantation in rats models of spinal cord injury, a systematic review. His area of interest is stem cell biology, tissue engineering, genetics, and neurodegenerative disorders. His future directions: he will be moving on to PhD in cellular and molecular biology or stem cell biology. His aim today is to provide basics on stem cells to the audience and the role of stem cells in medical research presently and the advanced research stem cells which are ongoing. He would like to make his audience understand about the potential and possibilities of stem cell research and help create new thoughts to individuals who wish to proceed in this field. Also, he has submitted several reports and submitted manuscripts. I do believe that he is indeed a great role model for our students. By saying this, I hand over the sessions to you. Thank you, sir.
Thank you, Hina. I appreciate the introduction. It's great speaking with all of you today. I would like to share my screen with you. 1 second. Can everyone see my screen? I hope everyone can see my screen. Yeah, as the host said earlier, today we're going to speak about stem cell research and its fundamentals. I'm very sure most of you are here to know what is stem cells and what are we doing with it so far. And half of you might be leaving your laptops or mobiles aside, thinking this might be a boring topic. I hope I can bring you people attention back to the screen. And without any delay, let's move on to the session.
Excuse me, sir.
Yeah.
May I interrupt?
It's okay. Tell me.
Yes. Could you please show your video? Switch on your
Here.
video. Thank you. Thank you, sir.
Yeah. So today, as I said earlier, today we're going to talk about stem cell research and its fundamentals. So when we look at the history, where are these stem cells coming from? But it dated back to 1868. It's almost 150 years. So you can imagine a 150 years of research, how much enormous amount of evidence and research has been conducted on these stem cells. And looking at these timeline, you can see there are a number of achievements, milestones. In 1909, there was blood stem cells first discovered by a Russian scientist, followed by a unsuccessful bone marrow transplant and a successful bone marrow transplant by Dr. Robert. So these events led to the events of creating this stem cell research. And the first and foremost milestone was deriving embryonic stem cells from mouse embryo in 1981, followed by discovery of a method to derive stem cells from human embryos. This is just a fair look on what all this history about. Coming to the main event, if you can see these two prominent scientists here, John B. Gordon and Shinya Yamanaka, these are like prominent researchers and stem cell biologists in this field who got Nobel Prize for their experiment. So what they, in simple words, what they did is they took the skin cells, reprogrammed it back to the stem cells. So it's like you can take the skin and create any kind of cell, whichever you desire, like heart cell or neural cell or kidney cell. You can create almost any kind of tissue out of it. So just by taking a skin cell, they were able to reprogram it into pluripotent stem cells. So for this, they got the Nobel Prize in 2012. So this is the paper article, which if you want to go back and read, induction of pluripotent stem cells from mouse embryonic and adult fibroblast cultures by defined factors. So what they did is they found the responsible factors which are necessary to convert normal cells into stem cells and use them to create any kind of tissue. So if you want to have a look, you can have a look, just take this title and you can get the information about it. Moving on to the next slide. I really want to show, it's not needed here, but I really want to explain about this so-called Hela cells. So these are first obtained from a female lady, female person with cervical cancer. In 1951, it was obtained. So these are the first successful proven human cell line. From this, they expand the cells and conduct research. So almost 80,000 of scientific articles and studies has been conducted, particularly these Hela cells. So these are like a superhero of the stem cells. So far, number of human cell lines has been developed from this single cell, which is trying to crawl here, if you can see it on the screen. So moving on to the next. So what are these? So I've been telling about stem cells and history. So what are these stem cells? Stem cells are nothing but a cell with two fundamental properties. One is self-renewal. So it is having the capacity to renew itself. So it will make itself a copy of it, like number of it. And the other property is differentiation. It can differentiate into any type of cell. For example, if you have a look at this, so this is what basically I'm trying to say. A stem cell can replicate into number of cells, like renewing, like creating a copy, creating a clone. And when it comes to differentiation, it can turn either, you can see it here, a blood vessel, a blood cells, or heart cells, or neurons, or skeletal muscle. So basically, it can convert into any kind of. To put it in simple way, take a student who is about to take his degree. So he can be a doctor, he can be a lawyer, he can be a engineer, but it depends on what he's choosing, right? So the same way, this stem cell can be anything, depending on what kind of factors we are giving it to get converted into that particular cell. So moving on to the next slide. So as I said, differentiation and self-renewal are the properties of a stem cell. When it comes to human, that too particularly scientists, curiosity is the common property of a scientist. So when they came to know about the stem cell, they started understanding it. They tried to learn more about it. So they started to know what kind of stem cells exist, from where they come, and what kind of conditions are needed to maintain those particular stem cells. And also they were able to understand that these stem cells have the potential to cure a disease. So far, only minor research has been done, which is successful enough to apply in clinical settings. So I will discuss that later in the slides. And also these stem cells are also used for drug development and toxicology studies as well. Moving on to the next slide. So their curiosity has been successful. So they were able to understand what kind of stem cells exist. Most of you might have seen these jargon in either pharmacology subject or human anatomy HAP subject. So totipotent, pluripotent, multipotent, oligopotent, and unipotent. So these are the types of stem cells which you can find in the whole body. And mostly in current research, they use pluripotent and multipotent stem cells and derive oligopotent and unipotent from the above things. So commercially available are mesenchymal stem cells, hematopoietic stem cells, neural and induced pluripotent stem cells. So these can give rise to particular cell, but after inducing particular factors into them. So that's how they classified potency. Yeah. Sorry. Okay. Moving on to the next slide. So this is the pictorial representation of what I said earlier. So totipotent is nothing but blastomere. So all of you know about blastomere. It is like a product, development of zygote to blastocyst and blastomere. So that is like totipotent. It is having the capability of developing the whole organism. And coming to the pluripotent, it is nothing but embryonic stem cells or induced pluripotent stem cells, which can create specific tissue or cell, but not the whole organism. They can create any kind of somatic cell, but not the reproductive system like amniotic sac or germ cells related to those areas. So these are only like creating somatic cells. When we come to multipotent, so these are derived from embryonic stem cells or induced pluripotent stem cells. So mesenchymal stem cells can create bone, cartilage, muscle, or skin. And similarly, hematopoietic stem cells, the name itself tells that it is related to blood, and obviously it's gonna it is having the capacity to develop any kind of blood cell. And similarly to the neural stem cells, the name defines that it's going to generate neurons and oligodendrocytes and astrocytes. So these are all the common things which can be happening with the stem cells. But one particular thing that made me also to so these particular somatic cells are nothing but skin or hair follicle or any kind of cell which is seen in an adult body. So this particular thing caught my attention. That's how I went into this field. So when these somatic cells, as I said earlier, reprogrammed back into the pluripotent stem cell. So these are the reprogramming factors which convert these somatic cells back to the induced pluripotent stem cells. It's like sending a cell back to the past. So it's like a time travel for the cell, and it can change into any kind of tissue. For example, I took the muscle cell, I added the reprogramming factors, and it converts to induced pluripotent stem cells. From there, I can create any kind of tissue and use it for research. So it's like a multi-dimensional multitude of research possibilities with this particular iPSCs. So that's where I got struck with the idea of conducting research on neurodegenerative diseases using iPSCs. Moving on to the next slide. So we learned so far that stem cells are these, so and so types are exist, but where do we get them? What is the primary source for stem cells? So you can find stem cells almost in every part of the body: eye, teeth, lung, liver, kidney, skin. Almost every part of the body contains stem cells. Only thing is they are like unspecified cells and they're inactive, and it is difficult to obtain most of the places. So only some specified places, for example, bone marrow or kidneys, like major organs are utilized to harvest these stem cells. Not only these, recently they also came to know not only organs or tissues, but body fluids also are able to generate these stem cells. They also having these stem cells. So this is one article, one study conducted that where they came to know that sweat is having stem cells. Who will imagine when we are sweating and we can just take a drop of sweat and create tissues? No one gonna expect that, like, oh, we can take sweat and create tissues out of it. No one have guessed that, right? So this study where they understood that even sweat contains stem cells which can be used, which can be harvested, which can be isolated to conduct research. Not only sweat, take saliva. Exosomes are nothing but factors released by stem cells. So even saliva, it is able to promote angiogenesis and cutaneous wound healing. So even saliva, while we are kids, we used to do this thing, taking saliva and rubbing on the wound, thinking that it will heal. And most of the time it's true. And now now we know why it is true. Similarly, even the urine. So whenever you pass the urine, you think it is just waste product. So, but for scientists, they find like it's like a treasure for them. It must be funny, but it's like a treasure for them. So they find number of things in that particular thing. So in this particular study, they derived stem cells from urine and they used them for renal ischemic perfusion injury in a rat model. They tried to treat it and it's successful in rat models. Yeah, in rat models. So far, whatever the research done with the stem cells, it's in animal model or cellular model, and it takes extensive research to bring that to the clinical setting and apply it on the patients directly. So we are far from that scenario. Coming to the isolation. So we know the source, but how do we get them? We know we know that the stem cells are in my skin, but how do I get them out? How do I take them out in a perfectly good condition so that I can use it for research? So that's where this process comes. So you take the tissue, you mechanically slice it up into pieces, and we use collagenase. It's a digestive enzyme which cracks the tissue, and we separate the important cells, the so-called supernatant liquid will be separated, and the pellet resuspension. Here you can see the small pellet. So this is the clump of cells which are at the which is at the end of this ampule. So these are resuspended into the petri dishes or these cubic plates, and they are incubated to absorb these cells. So from a small tissue, they're going to do this process and they get the cells, and they use these cells for commercial and medical purposes. They conduct research, they use it for therapies. There are like, I have seen many news, like most of the clinics in USA or Europe, they use these stem cells for the treatment without any FDA approval. So they just use plainly. So I just want to give a small, slight hallmark warning here that most of the clinics which you find that they're going to provide stem cell treatment, either it is like worthless or either it is like a fake thing. So not all, but few, because I will explain available treatments, available stem cell therapies in the upcoming slides so that you can know how to differentiate which is fake and which is a real one. So coming to the sulfate. So now you got the cells, you harvested them, you isolated, and you got them on the plate. So how do you make them into necessary cells? I mean, well, now you're conducting an experiment on, for example, brain. So you need neural cells. So how do you guide those stem cells to become a neural cells? How do you make them into oligodendrocytes? How do you make them into astrocytes? So that is called cell fate. So a stem cell can be anything. Like we can make it into a neural cell, epithelial cell, immune, muscle, heart. We can make it into any kind of cell. But how do we do it? So that is where comes stem cell signaling. So we need to provide some signals, some directions to the stem cell so that it can be converted into our respective tissue or cell. So for example, take this thing. We know that self-renewal and differentiation are the two key properties. So, but how do we know that these gonna be in proper direction? How do we know that we are getting the cell tissue we need? I need neurous tissue, but how do I know that I'm getting neurous tissue? So these are the transcription factors which help direct them into particular direction so that they can be converted into specific tissue. So these transcription factors, epigenetic modifiers, and signaling pathways, these together, all together, direct these stem cells to convert into specific cell or tissue. So as I mentioned earlier about Shinya Yamanaka, so these are the factors they established. They found that these 4 factors are involved in the maintenance of pluripotent stem cells. So this is the breakthrough they did, and for this work they got Nobel Prize. So, and of all these 4 also, I highlighted 3, OCT4, SOX2, and NANOG as commonly considered pluripotency factors, because when we are having such advanced research, there will be some drawbacks, and that one of the drawback is this factor c-MYC. So it is also found in cancer, where it induces cancer, like uncontrolled proliferation of cells. So this is excluded out of this list, and they use these 3 to induce stem cells to differentiate into specific tissue. Moving on to the next slide. So I mentioned that we have types of signaling. So what are all the types of signal? You might have heard this VNT signaling, TGF, tumor growth factor, and fibroblast growth factor. All these signalings we have studied in pharmacology and HAP. We have got that jargon in HAP as well. So what are all these things? What do they do? So they involve in this cellular proliferation. It's nothing but expansion of cells and adhesion. So we inject the stem cells into the body, and how do we know that they stick to the particular location? So these things signal them, they direct them to that particular location and help them to proliferate and attach, and help them to migrate to that particular location. Not only these things, there are like multiple signaling pathways. I just mentioned the prominent ones. So these are the prominent ones which help them in migration, differentiation, and survival. So these are the main signaling pathways which will help in stem cell differentiation, migration. So moving on to the next slide, stem cell niche. So we know our body is the source of stem cells, but inside our body, where do they lie? I mean, where do they stay? What is their home? So that is called as stem cell niche. So they stay here in a specialized microenvironment where they attach to the extracellular matrix and the normal cell tissue, and they help them proliferate and control the number. They regulate the number through extrinsic factors. So these factors control the stem cell. If there is no control on the stem cell, you got an injury. The stem cells reach the injury site, they try to heal it. And if there is no regulation, you'll get hyperplasia or like cancer. So that is how there are a number of factors which will regulate the stem cell number, proliferation, and its determination. So this is very important as well, the niche and its extrinsic factors. Moving on to the next slide. So we spoke about all these things. We have our history of stem cells. We studied stem cells, from where they will appear, what is the source, how do we isolate them, how do we direct them. No, we have all the stem cells in our hand. So what do we do with them? So that's this first thing. So the research to study rare cell types. Most of the scientists still argue that we don't know completely about our own body. Still, we don't know number of cells still there in the body. So these stem cells help to study these rare cell types. Not only that, disease modeling. Most of the disease, they don't have proper treatment. We just know their pathology, but we can't do anything about it. So what we do is take the stem cells, create a disease model. It can be 2 types, either cellular model in a petri dish or an animal model where you induce this particular pathology in the mouse model and use these stem cells to create the same and microenvironment and try to treat them. So for example, neurodegenerative diseases, Parkinson's, Alzheimer's. So in these kind of diseases, we don't know how to cure them properly. I mean, in the full extent where the quality of life is the fullest. So at that moment, we take these stem cells to model the Alzheimer's disease and try to study its properties, its pathophysiology, and how to treat it. So that's how these stem cells used in disease modeling. Going on to the next, drug screening. This is one of the prominent method to understand how drugs act as well. So as I said, these cells can be replicated as a particular tissue. So take the stem cells, make them into neural tissue, and apply the drugs which you create for neurodegenerative diseases. You're going to inject that drug into that particular tissue, and it will act. Then they will study the properties of how it is affecting, how it is reacting. So that's how they know this particular drug will cure this particular disease. This particular drug acts in this way, so this tissue is reacting in such a way. So we do this drug screening, pharmacological tests, so that we can better understand the drugs. So the last one is transplantation therapies. So you might have heard bone marrow transplant, which is for leukemia. So this is a kind of transplantation where you transplant stem cells to cure the disease. Likewise, there are a number of methods in which these stem cells can be utilized. For example, cell therapies, toxicology screening, drug delivery, drug validation. So there are a number of opportunities which can be utilized by these stem cells. Moving on to this next slide. So these are the things which I mentioned earlier. We can get stem cell either from embryo or a patient and convert them into necessary stem cells and use them for either regenerative medicine, drug safety testing, disease modeling, or personalized medicine. So there is a patient, he's having cancer. We take the cancer cells and use them to create a disease model. So we can create that particular cancer in a petri dish, and we can study cancer drugs on that particular disease model. So if you remember, I mentioned about Hela cells. So this is how it happened. They took the cancer tissue from the lady and they isolated the stem cells. They created the disease model with the same. So that's how they started studying the cancer through this cellular modeling. So in the similar way, they can take any tissue, like normal tissue, and obtain functional cells and convert them into stem cells and use them in regenerative medicine. And there are some instances, there are some studies where they even created pea-sized brain or a beating heart in a petri dish. So they took stem cells of heart and they created a beating heart inside a petri dish. It was able to beat. It was able to like monitor the situation on. It can even give the ECG signals. So we are near to the research and yet so far from the successful application of those. So it's like a tricky thing when it comes to stem cells. I can say it's like hyper hope. So it can be either hype or just a small hope that we might be successful. So that's the way stem cells are considered nowadays. And moving on to the next slide. So what are the basic applications? We did everything. We got isolated stem cells. We are able to convert them into necessary specific cell type or specific tissue. So how these stem cells work? I mean, how this therapy work? As I mentioned earlier, we take the stem cells, transplant them, and growth factors direct them what to do, and they will differentiate into the tissue surrounding. Sorry. Yeah. Back to the screen. So this is the small process of how visual representation. We'll take the diseased tissue from the patient and we isolate the cells, cultivate them, expand them, and use them for the development of tissue and implant back to the patient. So this is like a basic process. It might look simple in this particular cycle, but it's not as simple as this. So after taking biopsy to isolate cells, it's going to take 2 months. Then to cultivate the cells, it's going to require extensive support with the laboratories. And to maintain the cell proliferation, it's going to cost lakhs and crores of money. So, but at the same time, the outcome is also equally beneficial. So it is able to save a human life just by taking a small cell out of tissue. So the drawbacks and the advantages are balanced here. Coming to the next point. So can doctors use stem cells to treat patients? I will say yes, because like from the past 40 years, there are a number of peripheral blood cell transplantations and bone marrow transplantations happening for these cancers, leukemia, lymphoma, and many others. So I will say yes, we can use stem cells for the treatment and doctors can use them. Even in Bangalore, there are multiple hospitals like Plexus Stem Cell Care where they use stem cells to cure these neurodegenerative diseases and spinal cord injuries as well. So there are many other diseases which are targeted by this stem cell therapy, like diabetes, spinal cord injury, muscular dystrophy. As I have mentioned everything on the screen, you can look at them, the vision and hearing loss. So most of these diseases can be cured, at least in animal model so far. And moving on to the next slide. So these are like almost every disease can be covered with stem cells. We can obtain any kind of cells from this particular damaged tissue, create a disease model, and try to study the disease and try to get a treatment, like optimal treatment, which will be effective enough to cure that particular disease. So you can see there are a number of diseases here. Almost you have studied almost everything here. And you can see the one here, bone marrow transplantation, which is currently established. As I said, it is done so for like almost 40 years, they're using it for the cancer. So number of diseases can be cured if we are able to harvest, if we are able to harness the power of stem cells in this particular field. So I will just give you an outline of spinal cord injury as an example for today. So they have taken mesenchymal stem cells. As I said, these are multipotent stem cells. They use these in spinal cord injury to improve the functional recovery. This is just one study. There are like lakhs of studies on spinal cord injury, which will help them recover functionally. And this is one more study where they use neural stem cells to increase the oligodendral and neural differentiation in spinal cord injury. As I said, we can use particular growth factors and stem cells to differentiate them into proper way. Like we can alter the course of that particular tissue, damaged tissue. Similarly, we have one more, transplantation of neural stem cells in alginate scaffold for spinal cord injury. So likewise, there are a number of studies where they use stem cells to cure particular disease in animal model and cellular model. And these spinal cord injury patients were also treated. There are a number of articles and studies conducted on humans as well for spinal cord injury, because once if you are affected with spinal cord injury, it's like for a lifetime you will be like bedridden. So it is difficult to cure this particular treat this particular spinal cord injury, and these stem cells are giving a promise that they can help recover the function. They can help to cure this particular spinal cord injury. Moving on to the stem cells in regeneration. So most of you have seen these really old movies where villain will be there, superhero will be there, and the villain, superhero will ampute the villain's hand and he will regenerate one more. He will ampute again and he will regenerate again one more. So it's like a continuous cycle. But how come he does that? I mean, how is it even possible to get regeneration? Yes, it is possible. Even in humans, regeneration is possible, but it's been suppressed. And you can see if you get a cut, like a small knife cut or something, your wound will be healed. How come it's healed? It's just nothing but regenerating the lost skin, right? It's just closing the gap by sending the stem cells. So in the same way, like villain way, these stem cells help in regeneration as well. I want to give one particular example. If you ever heard of the word planaria. So this is planaria, where these are like masters of regeneration. So whenever you dissect, I mean, whenever you cut planaria in between, like for example, this in image A, we can cut the planaria in horizontal way, and each part is able to develop its own body. You can see it. The first part is able to have its own eyes and its own tail. The middle part is able to develop eyes and tail as well. The tail part is able to develop head as well with eyes. So how is it even possible? They are able to develop eyes as well. Just taking a small part of this planaria, we're able to generate a new organism. And the surprising thing in here is they're able to retain their memories as well. So when you cut this particular planaria into 3 pieces, each piece have a copy of memories from the main organism. So they're able to recall the memories as well. So how is it possible? Why is it not possible in humans? Why only planarians? So there are a number of questions which can be answered by studying stem cells, by conducting research on stem cells. So this particular white thing, if you can see it on this particular piece of the body, slice of the body, this is called blastema, the place where stem cells accumulate and help it for regeneration. So that's how they regenerate these eyes and tail, everything. Not only in that way, you can either cut it in a mid-sagittal plane or horizontal or uneven, or even take a small piece of the body. It can regenerate a whole organism. So this is like one of the most powerful regeneration model, which we study in stem cells to understand how this can be applied in humans, why it's not happening in humans, why in planarians. So this is the experiment which was conducted by me back in UK, where I took these 3 planarians and I made them into two parts, head and tail. And I checked the effect of retinoic acid, how it affects. Most of you know the retinoic acid is retinol, nothing but vitamin A. It helps in regeneration. So we tried to study up to what extent retinoic acid can help these planarians grow their body back. So you can see this is the lowest concentration, 20, and here is 200 molars. So we use this to study, and surprisingly, the lowest concentration is able to develop quicker. So you can see the tail part in head part is developing tail and tail part is developing the head. So likewise, we can direct the regeneration by using these kind of factors. For example, in this case, retinoic acid. So likewise, I will give one more example. So this is called scratch assay, which we did in back in UK. So this is nothing but wound healing. Earlier I mentioned if we have a cut with a small knife, we'll be having a small cut which will be closed in eventually. So this particular thing is nothing but the wound. So I took a knife and sliced the cells which were implanted on the petri dish. So this is the line, or you can call it as wound. So this particular condition, this is control, this is a test. So we are testing how mesenchymal stem cells were able to increase or are able to improve the migration capacity as well as healing capacity of the skin. So here you can see within 50 hours, this particular condition is able to close the wound. You can see gradually it is getting closed all the way down. Sorry. And when you see in the control, there was a small minor gap which was not able to heal the wound properly within 2 days. So when we applied these stem cells, it was able to heal properly and quickly. So this is how stem cells can be used in angiogenesis and cutaneous regeneration, where we can heal the chronic wounds, for example, diabetic ulcers. Diabetic foot ulcers. So on. So there are a number of chronic wounds which we can cure or which we can improve the treatment by using stem cells. So that is one example as well. When coming to the stem cell research, there are a number of obstacles. Obviously, when there is a cutting edge research, it will come with setbacks as well. So one is how to find the right type of stem cell to use. So we got number of types like mesenchymal stem cells, hematopoietic stem cells, or neural stem cells. So which one to use? So that's one question which is bothering everyone. And they found the answer as well, because they were able to take the induced pluripotent stem cells. As I said, they got this Nobel Prize for that. So that's how the problem got solved. So they take this iPSCs, they differentiate into a specific cell. Coming to the next one, how to put stem cells in the right place. So for example, I got spinal cord injury, but how do I send the stem cells to that particular spot? How do they know that this is the injury site? How do they know? So that's where the homing of stem cells, the migration comes in. We can give stem cells either by intravenous, or we can inject them directly to the injury site. So these are two methods. We can use subcutaneous or multiple injection methods as well. So I'm just taking intravenous an example. So when we give stem cells through intravenous, they're going to travel all the way through the body, and they will reach the site of injury, and they will help the injury to heal. In the similar way, we can give them on the site, like directly cut open the body and place the stem cells on the injury, and it's going to heal. So similarly, what is the right place? So this is one more question as well. And will the stem cells perform the desired function? So I took the stem cells, I kept it in my spinal cord. I injected them into spinal cord injury. Now I will sit and wait to heal that particular injury. But how do I know it is working in the desired way? How do I know it is developing only spinal cord, that oligodendrocytes or neural tissue which is needed, or bone tissue which is needed? What if it developed into a hair? What if it developed into a heart cell? How do I know? So that's how we monitor. That's how we use that transcription factors and growth factors to direct them into that particular cell type so that it can show the desired function. That is one. And coming to the next, development of differentiation protocols. So we have these stem cells and we have the growth factors. How do we know which growth factor will direct stem cell into particular tissue? So what are the protocols to conduct this research? How do we differentiate them? So these are all experimental protocols. We call them experimental protocols, where they create their own protocol, apply it. If it is successful, they're going to use it for the research. If it is not successful, they will study it, why it is not successful, what is holding them back. So they're going to do this experimental protocol studies as well. And coming to whether it is safe and effective. We know something called immune system, which will reject outside foreign bodies, whatever. So when we take stem cells from another person, autonomous, so it will be rejected. But what if we take stem cells from our own body, convert them into a desired tissue and inject back to us? So that's how the problem will be solved. So autologous stem cells, that is called autologous stem cells, where we take stem cells from our own body, convert them into desired tissue and use them back to the same person to treat the disease. So it is effective at sometimes, it is safe in sometimes as well. So these stem cells sometimes can be unpredictable and they can form cancer as well. That is called teratoma. So for this particular thing, they started to study their abilities by using a quality control assay, where they will understand whether this particular tissue is able to develop cancer or not. That's so-called teratoma. It can be termed as cancer and it can be termed as teratoma, where it can produce all kind of tissue. There is one situation where they used the stem cell therapy, and there was a formation of teratoma where teeth, hair, and all kind of skin, everything was formed in one particular place. I was not able to provide that image. It might be disturbing, so I didn't provide the image, but I hope you can search the teratoma image if you want to. Coming to the ethical debate. So we know totipotent stem cells are obtained from blastomere. It's nothing but life. So when you harvest embryonic stem cells from this particular blastocyst, it's nothing but taking like life from the zygote, from the blastocyst. So it is unethical to do this. It's like nothing but murder. So embryonic stem cell research is banned back in days so that they won't harm the human life. It just nothing but it even creates a commercial market for human cells, where they will try to what is a medical mafia, where they will take the human cells. It's almost like devalues the life. There is no more value. They can take the human cells, they can create the clones, and they can reproduce the same. So it doesn't create balance in life. So this devalues life. So there are a number of ethical debates which is surrounding these stem cells. So for this reason, they kept a ban on embryonic stem cells and they started using adult stem cells. As I spoke earlier, the multipotent stem cells, mesenchymal stem cells, hematopoietic, neural. So these are the cells which are used so far so that they can stop using embryonic stem cells. And moving on to the final conclusion. So far, stem cell research has been very rapidly changing the way we look at the disease and its treatment. Not only this, most of the traditional treatments available for the diseases so far, they can be replaced with stem cell therapy in future, not near, but we are doing this extensive research where we can get to that particular scenario where we can replace the traditional treatment with the stem cells. Not only this, from past few decades, there are a number of stem cell therapies which are conducted experimentally on humans, which are taken by the ethical committees as well. There is no illegal thing, but it's a legal thing where they use stem cell therapy and they made it like a game changer. They used this bone marrow transplantation to cure the leukemia. So there are a number of instances where the stem cell therapy proved that it can be a promising future. So that's how these stem cells involve in medicine so that they can help future to model itself in a better place. I hope you understood all the points which I mentioned so far. If you are people having any questions, I can answer them now. I'm giving the opportunity to you all so that you can ask some questions. If you want to drop the questions in the Dropbox, you can do it on the inbox as well. Thank you for the opportunity for listening as well.
Thank you, sir. So it was indeed an eye-opening session that you have provided us. Thank you, sir, once again.
Yeah.
And we have seen what are this, so far we have seen what are stem cells, its potency-based classification, its sources, its isolation of cells, and what do we do with it?
Yeah.
Its basic application in the industry and in the medical field.
Yeah.
And how it helps in various diseases. Also, the obstacle faced in the research and designing of stem cells and the ethics behind this. It was indeed a wonderful presentation, sir. Now we have a question and answer sessions, and the students are more than welcome to ask questions to Dr. Anil. He will reply to you. Any questions?
Yeah, you can have any kind of questions. If I'm in your place, I will be asking like a ton of questions actually. So particularly in you field, and obviously we'll be having a lot of doubts. Don't hesitate to ask. So it's up to you. I can see. So everyone is so clear enough to understand what is stem cell research then.
Guys, if you have questions, please do ask. Yes.
I can see a question here.
Yeah.
Can stem cell therapy able to treat cancer? So far, there is no approved therapy for the stem cell for the treatment of cancer. But back in UK, when I was studying, there is one PhD guy who is trying to do that. He used these cancer stem cells to study how to treat them. So there are ways. There are in theoretically yes, but in practical way, it's not yet possible. It's still the extensive research is going on. So I will say a straight answer, no, not yet. I hope that answer justifies your question. Any other questions? Thank you so much. I can still take on the questions if anyone are having any doubt. So as I mentioned, what diseases can cure? So as I mentioned, there are like a few cancers like leukemia. That is different to what the cancer transplantation for this particular thing. And also in few instances, they use stem cells for treating the spinal cord injury. As I showed the articles earlier, it was able to improve functionally. In that, there are human studies as well. If you want, I can send the links afterwards. So I can show what all the human studies done so far to treat this particular disease, so and so diseases as well. So how was your experience with stem cell research in UK? Yeah, pretty good, because most of the, if you see what kind of stem cells research was conducted in the whole world, top places will be like Japan, Korea, China, UK, and Canada, USA. So these are the top 5 places or 6 places which are very good for stem cell research. So I enjoyed my time in UK while I was conducting my stem cell research. I know there is this pandemic and the lockdown thing and going on, but still I was able to enjoy my research there. Negative effects, as I said, there is a possibility of teratoma formation. So we can be like able to control. Sometimes they will be uncontrollable because they are in they are unspecified cells, and we are giving some signals so that they can become the specified tissue. Yes, there are negative effects as well, like teratoma formation, or sometimes it won't be effective enough. It will be like not up to the mark as the medicines available in this market. So as I'm saying the same thing, this ADRs and stem cell therapy, so far, whatever there in here, this can be very tricky to understand the negative effects, because for example, this spinal cord injury, they are having a minimal effect when it is used in normal way, I mean, in intravenous way. But when it is used in surgical way, you need to cut open. So it's going to give the pain, and it's going to induce much more effects as well. What about psoriasis? As I said, almost every disease condition so far is being studied in a cellular model and animal model. So even the psoriasis, they have been studying in this animal model where they create this psoriasis as a disease model in the rat, and they are trying to study how these stem cells can affect on that particular psoriasis rat. Night blindness associated eye defect. Yeah, as I said, so far only we can count on fingers what are all the available therapies for stem cell therapies. So whichever the disease I can see in this chart, like night blindness, psoriasis, or cancer, all these are like in a development stage. They are still doing this extensive research so that they can get this particular outcome where they can cure this totally. So far, only animal studies and cellular studies has been done, which are giving promising results. I can say that they are giving promising results. They were able to cure that in that particular model, but they haven't brought it to the clinical setting yet. Is lifestyle disease can be cured with this therapy? I haven't heard about that particular thing, but I will get back to that lifestyle disease and I will let you know if you are happy to contact me again, because I'm not sure about that lifestyle disease. Does stem cell replace complete defective cells? So far, whenever I study or whenever I saw articles which are related to this, yes, almost like they are able to replace, but it needs extra support as well. So we need to add like supporting drugs, supporting therapy as well to completely replace these defective cells. For instance, if you take this injury, cut injury, wound healing, chronic wound healing, it can replace the totally defective cells, but still there is a scar formation. So that can be avoided. So there are such instances where we can't exactly tell that this can be reflected in the same way as we expected. And next, do we have this treatment if stem cell in Bangalore? Can you give me any contact? Yeah, there are 2-3 hospitals where they provide stem cell therapy, but it depends on which disease, because I know about this Plexus Stem Cell Hospital where they treat the neurodegenerative diseases. So we can contact that particular hospitals for stem cell therapy, but I'm not sure for which disease you are asking about, man. So any other questions? So do we have any other questions? Or retinal damage, can we go for this treatment? In India, I don't think so it's a good decision to take stem cell therapy, because most of the therapies in here are still experimental and not yet approved. Not only here, even in abroad, there are some places like Florida and Minnesota where they are doing this experimental stem cell therapy for almost all kind of diseases. So I suggest to look at those possibilities instead of looking it for in Bangalore or in India. But I don't think so it is approved yet. They just give the stem cells as a support so that it will enhance or it will improve the condition. Why stem cell transplant therapy is so much costly in treating cancer patients, and is there any way to reduce the cost of this therapy? As we know, whenever we try to harvest stem cells, it takes a lot of effort and a lot of manpower and cutting edge laboratories. So to maintain all these, obviously we need a lot of money. So it costs more for this particular reason. And not only that, because of the hype, because the stem cells were able to do this magic, people think that patients gonna come to them and they're gonna cash it out. So it's like a selfish kind of thing, which makes them it costly as well for treating cancer patients. Not only like commercial based, it's all like to make more money out of it. That's one thing, to be frank. So the main thing is it's because of the research conducted, the laboratories, everything to make it. Yeah. To reduce the cost of therapy, it takes few more years. The previously, for example, take PCR, the one which you are doing for COVID-19 test. So previously it takes like almost 2-3 days to conduct a PCR. Now it is taking only 3 hours, right? So the cost changed. Previously it's going to cost thousands of dollars. Now it's just like 1,200 rupees to conduct a PCR test. So similarly, right now we're in a stage where this stem cell therapy is costly, but we will reach a scenario where this will be cheap because we are able to understand the stem cells by conducting research. So this closes the gap between the actual scenario of what we are anticipating in future. Any other questions? Is stem cell therapy is able to treat genetic problem? If I repeat myself, so almost all diseases, even genetic problems, they are able to be treated in animal models and cellular models. So they have this petri dish cellular model of the disease or animal model where they do these experiments by genetic, by using CRISPR, by editing the genome in the stem cells as well. And they use these so-called exosomes or engineered mRNA, like the one which we got this vaccine, the COVID vaccine, which is nothing but mRNA thing. So they do all these things to treat the genetic problem as well. So I can strive for that these are still in the lab. They haven't reached it to the clinical setting. There might be few which I have missed that they might have reached to the clinical setting, but as far as I know, as far as according to my knowledge, they're still in the laboratory, not to the clinical setting. Yeah. Is stem cell helpful for neuronal ceroid lipofuscinous essential disease? I'm not sure about this particular disease, but I haven't heard actually, so I can't answer this particular question. I hope I can get back later with the same. Any other questions?
Any more questions? Okay. The session was very fascinating and it was very informative, sir. I thank everyone once again for being a part of this webinar, especially the students and all the faculty members. Especially I thank Dr. B.A. Vishwanath, who is the chairman and the backbone of our institution, as well as Dr. Priya Vishwanath, who is the vice chairperson. Now I call upon Mrs. Bharati, who is a faculty member of the Department of Pharmacology, to give the vote of thanks.
If I can speak 1 second. I also would like to thank our chairman, sir, and Dr. Venkatesan, sir, who approached me for this, and I'm very grateful for him and to the chairman, sir, for letting me take this session. I'm really happy that I'm able to do this with our college students and staff. Thank you so much.
Hi, Anil, such a remarkable session, very phenomenal. And, especially the student from a college giving a phenomenal and mind-boggling session. So we're very proud of you being here today. So I would like to propose a vote of thanks today. I express my sincere gratitude to all the participants who attended this webinar. Because of you guys, we could make this webinar a very successful one. And I would like to thank our chairman for continuously motivating and encouraging all the staff members and the students and the alumni members, because he is always a backbone for us to encourage such kind of webinars, not just from the staff, not just from the outside people, but also from the alumni members. I also thank our dynamic lady or vice principal of a college, Mrs. Suchitra, Professor Suchitra, for giving us an opportunity to all of us. And I also thank Dr. Venkatesan for identifying and then making the people to come and talk over in the session. And I thank Anil for giving such a beautiful session, very wonderful. And I know that, after listening to your session, each one of us after is extremely that you are the alumni of college. And it is so beautiful to see you. It is such a nice session with a beautiful explanation. And I also thank all the staff members, all the teachers for continuously helping us in organizing this webinar. Thank you so much. And I hope you enjoyed this webinar. Thank you so much. I thank Reenu as well for and wish every day. And I know that, by listening to this session, I think, a lot of students from a college can ignite their minds and probably they can enter into this research. So it's a great start to have. Thank you very much.
Thank you, ma'am.
Thank you, ma'am. So everybody, I thank you once again for attending this session. I hope you have a happy World's Pharmacist Day. Thank you all once again.